I know it's the end of the day, so I'm going to try to make it not too technical, but then actually the scope of the talk is a little bit technical, so I'll try to kind of keep it out of much jargon as possible. I think that's something that I can owe you. So this is generally to do with feature selection, right? There are many different ways of doing feature selection, and if you ask 10 people, you'll get 10 different ways of doing it. So the main thing here is that I wanted to go up with one of my methods, which I had come up with as part of my PhD, which I did in astrophysics. So essentially, some history back there, I was actually doing my theoretical astrophysics in studying black holes back in 2012, and I was working with one of the current Nobel laureates, actually. And the thing is, at that point, we lost funding because they didn't find any gravitational waves. So gravitational waves is one of the things which is postulated by Einstein's theory of general relativity. And so then at that point, I decided actually it was time for me to move on to something more practical. So I decided to move on to applied machine learning, and applied machine learning has a lot of scope in observational astronomy. So that's something that I wanted to actually put out one of my first couple of papers in this subject. And this entire talk is based on that paper. But then this genetic algorithm that I came up with is something that is applicable to any field because it's just a feature selection method that I'm going to talk about. And I have actually successfully employed this as part of a lot of different projects that I like. My areas of interest actually moved from astronomy. So I did my PhD in astronomy and astrophysics, and after that I moved on to finance. I was with Goldman Sachs for a bit, and then I worked for a hedge fund as a quant researcher before I moved to Flipkart as a staff decision scientist. So basically I'm a data scientist who works on analytics problems. All the way from pricing, selection, supply chain optimization, and so on. So you can see how ubiquitous something like this can be in all those fields, because one of the most important things that I find is if you can do feature selection and feature engineering on a large enough set of data which subsumes all of your, say, independent variables, then you pretty much can do anything you want. So that's kind of the ideology I'm going to move forward with. So this particular talk is only about feature selection, not feature engineering. So you're assuming that you have a large number of features. You want to say, okay, how can I best choose a small subset of features which best represents my so-called Y variable as such. So let's go on with this. So this is, so my paper is actually available in this link. So I think the presentation will be shared afterward. So you could have a look at that. It was published in the Astrophysical Journal a couple of years ago. And it was done in conjunction with the University of Maryland, NASA, and one of the telescopes in Hawaii called the Pan-STARRS telescope. So I'll briefly go over the data, what kind of data we're using. So because this is astronomy, that you may or may not have seen a lot of these things, but you've definitely heard of them, at least in passing mentions. The feature selection methodologies that are available out there, that will be the next part. Does pointer work? Pointer doesn't work, okay, no worries. Then I'm going to go over something called SVMs. I'm sure you guys would have heard of SVMs, support vector machines. So I'm going to use a feature selector in conjunction with a regressor, which is support vector machines in this particular case. And I'm going to solve two problems, one for classification, one on regression. So the good thing about SVMs is that it's kind of known for both really well. And I'm going to tell you why I'm going to use a GA along with an SVM and not just an SVM, for example, right? So why genetic algorithms? And then on top of that, there is a layer I'm going to add on top of that. It's called a multidimensional covariance map, which is basically my addition on top of the vanilla genetic algorithm that you have, which drastically improves the convergence rate. So essentially, genetic algorithms are known to be quite inefficient in terms of convergence. I'm going to reduce the factor of convergence by a factor of 100 or so. And then the future work, genetic programming, is something of a discussion that I wanted to have, but I think that's something I'll leave that for the time of questions, because I think that's slightly out of the scope of this particular talk. So the data. So there are two different types of data that we're looking at, right? One is, you look at an image like this, for example, right? You look at an image like this. This is your Hubble Space Telescope's view of the universe. What it does is it basically focuses on one part of the sky. It takes an image, it integrates it over a period of time, takes an image of stars and galaxies. So the first question is, you have a lot of things out there. What is a star and what is a galaxy? It's very difficult to do. Now this problem is very difficult to solve because the thing is, it's a very noisy problem. Secondly, you have 100 different telescopes basically telling you that this is a star, this is a galaxy, or it's just taking measurements of the same thing. It's not even telling you whether it's a star or a galaxy. The question is, how do you actually separate them out? So you have 1,000 different features from 100 different telescopes. You want to use all of them to come up with one classification system for stars and galaxies. That's number one. The second problem is that there are lots of distant quasars. You know, in astronomy, quasar is a distant galaxy which is emitting a lot of light because it actually has a black hole in the center of the galaxy, which is emitting light because it's eating up gas and emitting radiation. So that is visible across, say, thousands of parsecs. You know, or rather, in layman terms, it's light years, right? A couple of thousands of light years away, hundreds of thousands of light years or millions of light years away. And you want to actually understand, okay, how far something is. Now, the usual way of doing something like this is that you will take a spectrum of a line. For example, you'll take a spectrum, if you remember 12th standard chemistry, you have something called a spectrum of a line, basically located at a particular frequency. And if you actually Doppler shift that line, essentially because you're taking it away or bringing it closer, you basically will move the frequency by a bit. And based on how much it's shifted, you essentially get an idea of how fast it's moving away from you. And there is this equation which basically relates the speed of something moving away from you or towards you and the distance between yourself and that object. So essentially, you can get an idea of how far these quasars are based on this very complicated process. So I said, okay, well, you know what? You can't take spectrum for like 3 million galaxies out there. So is there a cheaper way to do this? In the sense that you have a lot of observations from hundreds of telescopes, you have thousands of variables, you know the distances to a couple of thousands of galaxies, can you use that to train a machine? So now what you actually have in hand is you have these two different problems, and what I suggested we do is we need feature selection on top of SVM, so some kind of regressor which is capable of doing this, but you can't just put 1,000 variables in SVM, it'll just die. There are two reasons why it'll die. First of all, the convergence timescale is going to be really long. And secondly, it's subject to noise. All these astronomical observations have a lot of noise. So when you put noise along with SVMs and you assume that the convex optimization is going to come up with the right solution for you, you're wrong. It's definitely going to actually factor the noise in and going to come up with a solution which is accounting for that noise, which you don't want. So essentially, how do you account for a reduced subset which is going to explain not only your star-galaxy classification in one case, but also your distances to the galaxies in the other. So these are the two wide problems, right? But the common feature in these two problems is basically that we just have 1,000 variables, and we want to come up with one Y variable, a solution to one Y variable, and we want to know what is the smallest subset of variables which can explain that. That's common across all domains that you've seen so far, right? So at any given point, please feel free to stop me and ask me questions as well, if it's not clear. By noise, I mean that, so essentially if you go further and further in the universe, right, what happens is that there is photometric noise, meaning which the counts of photons that you get from the distance actually is heteroscedastic. So something which is here has less noise as opposed to something which is slightly further away. So essentially when you end up using a variable which has a lot of noise and thousands of such variables, putting them in a regression, you're using a function of that noise as well and not just the variable to come up with an optimal solution. So I'm suggesting that removing an extra number of variables is going to help you come up with a better solution as opposed to having all 1,000 of them. So this is, yeah. So I'll give you some examples, right? Take this with a pinch of salt. I think you may or may not be able to read these axes here, but these are, for example, two variables I came up with which best define stars and galaxies, right? So essentially in this plot, the red is your stars and the blue is the galaxies, right? And the way I arrived at this was from 1,000 variables, I cut it down using my GA and an SVM, and basically came up with this classification. So essentially, this kind of indication where you have a separate class here, another class which looks like this is very classic radial basis function of SVMs, which is being applied on top of your existing variable spaces. So I'm sure all of you actually have heard of kernels, which are being applied to nonlinear spaces in order to linearize them and then fit an SVM on top of that, right? So it's very common stuff. So this kind of, when you look at two variables and plot them and say that, okay, here is one class and here is the other class, and it doesn't look like a clear straight line to you, that's an indication that an RBF kernel, for example, has been used, right? So here, this is something that I arrived at using my classification system for stars and galaxies. Here is basically your distance is essentially computed on your y-axis. Basically, this was the distance computed by the SVM, and this is the actual distance computed using spectrometer. So essentially using spectra of iron lines from those quasars, essentially you get an actual distance, and we got a regress distance using a subset of the variables using the GA and SVM here. So this is like both our experiments actually were very successful in achieving not only the distances galaxies, but also the star galaxy classification. So this is just an example of that. So let me go over feature selection methodologies. So typical nonlinear feature selection methodologies are not very common. I mean, generally, I think this is still a topic of debate in a lot of places, right? So for example, if it's linear feature selection, a lot of people will tell you, yeah, use an SVD, use PCA, use a lasso regression, use an elastic net. But what about nonlinear feature selection? It's not as clear-cut as that, right? So I'm going to outline a few things here. So essentially, you have the first layer, which is the method. The second layer is your challenge, right? So the first layer, go over the first layer. So essentially start off with kernel PCA, which is your first way of doing feature selection in a high-dimensional space. The problem with that is that you'll have to come up with a kernel choice. So kernel PCAs are where you actually have nonlinear data. You want to come up with features which best explain the variance of that data. So what you do is apply a kernel, transform it to an even higher dimensional space, and then you cut down the number of variables, right? But the problem is nobody is going to tell you what the kernel is. It's like it's not a given to you. So that's problem number one. Go ahead, please. SVM, you can actually, so SVM has a couple of different kernels that you can try out, and the thing is that you do have a iterative procedure for it, which is very well defined. So you don't have to worry about that a priori. But you're right, in the sense that, so for example, when I take the SVMs and I use it, I try all the possible kernels out, all the hyperparameters, I tune them very quickly. So I do have to do that. As far as I know, no. So it's like it depends. It's something which is data dependent. There are people who are attempting to do something like that, but in my view, it's almost a impossible problem to do directly. There is one way to do it, something called a genetic program, which will basically let you do one thing, which is take two x variables, for example, x1 and x2, and say x1 square plus x2 square is a good approximation to your y variable, as opposed to just x1, x2, for example. So you need to use a GP, and GPs are very different, right? So it's not the way in the process of using kernels. It's not the same as the process of using GPs. It's two different things. So in my view, you finding the right kernel is not an obvious thing. So we kind of typically tend to stick to maybe a RBF or maybe a sigmoid or things like that, which are much simpler to actually mathematically define and have clear-cut solutions as opposed to things which are not clear-cut, right? So that's problem number one here, right? Problem number two is generating embeddings using deep learning, right? So this is something that a lot of people do. You can actually use autoencoders, for example, generate these. So autoencoders are basically you have 1,000 inputs, the same 1,000 inputs as outputs, and you want to basically reduce the dimensionality in between. So it's like basically taking a huge pipe of water, passing it through a small funnel, and then taking it out again in a huge pipe, and seeing how much information you can retrieve from that. So the problem with autoencoders, again, is what is your network choice going to be? So depending on what your network choice is, how many hidden layers you have, what kind of activation functions you use, that's going to give you another issue, right? Third one is evolutionary, GAs and GPs. Now, genetic algorithms is what we focus on. The difference between genetic algorithms and genetic programming is actually significant. Genetic algorithms is only applied on the variables that you already have. Genetic programming actually defines functional transformations on top of your existing variables so that you need to optimize a combination of variable plus function. So for example, instead of using x1, you can say that x1 square x2 is a new variable that I'm going to create, or integration of x1 over a period of time is another variable, or some random function, which is actually topologically almost impossible using a machine, can be created. That's genetic programming. So we are not going to delve into genetic programming in this talk. It's something of a research interest of mine, but I think we don't have enough time for that. So the problem with GAs is that evaluating the fitness function for each combination of variables that you choose is quite computationally complex. It's actually going to take a lot of time. So if you have something like a simple correlation function, so for example, you take a function of x1, x2, x3, and that function basically is x1 plus x2 plus x3. Simple. You take a correlation with your y variable, great. That's as simple as it gets. But the thing is, usually what ends up happening is you use an SVM or a random forest on top of that. It's going to require more time to actually execute. So that's kind of the challenge when it comes to GAs. So I dealt with the challenge a little bit differently, where I actually took a subset of the data and I parallelized it over 1,000 cores. So I didn't have a problem over that. But still, it's a problem. It's a very tangible problem. The last one is using mutual information. So in one of my earlier talks, I had actually given about using a mutual information matrix of 1,000 by 1,000 matrix, which you actually create, and then using a Markov chain Monte Carlo to design thresholds between the X variable correlations and between the X and Y variables, right? The problem with mutual information-based methods is the binning becomes quite a challenge, because each variable, depending on how it's binned, your mutual information can actually significantly change. So that's, again, I'm not coding these as actual problems, but I'm just saying that these are your challenges, right? So we are going to go forward with the evolutionary GAs and GA method, which uses a fitness function at this point. This thing doesn't work. Oh, yeah. So this is basically I want to give a brief introduction to SVMs. SVMs are basically where you consider a maximum margin hyperplane, which divides two regions. So in the classification example, you basically have this to be your hyperplane, and essentially all your plus classes on this side, your minus classes on this side. In your regression example, you basically have all of your regressants, or whatever you're trying to regress, basically fall within this margin. So that's kind of the difference between your classification and regression. But SVMs can actually do both very successfully depending on how you place your constraints. So that's kind of a very simple modification to constraints, and you have a whole different convex optimization problem that you're solving. So on top of that, you basically can do one thing, which is that you have your input space, which is, say, n-dimensional. What you do is you transform it into a higher dimensional space using a kernel, and then you draw your maximum margin hyperplane, or you do your regression. So this is like the kernel SVM that I'm talking about, right? And as you rightly said, kernels basically you need to figure out what the kernels are, what the kernel parameters are, and so on. So that's kind of, since it's baked into a regular SVM, I kind of can optimize those parameters very easily, right? So why genetic algorithms? So consider independent variables from just x1 to x10, right? You have 10 variables. And I want to make this a simple problem to start with, so that we don't complicate it with 1,000 variables. And what subset of variables given a regressor, so basically subset of x, and you take the function of that, best represents y, right? So you want to find, let's say that subset is x1, x2, x3, for example, right? You want to be able to find that subset which best represents y, right? So you don't want to overfit your data, right? But you want to explain as much variance as possible, right? That's always the case in machine learning, right? So some bias variance trade-off and things like that. So the thing is that the goal remains the same, that you basically want to find the smallest possible subset which best represents your y, right? Where is it most useful? So why would I use genetic algorithms as opposed to anything else? Where noise can significantly skew results and dimensionality is really large, right? That's where GAs really excel. That's number one. And secondly, you can use GAs to optimize a correlated objective function. So what I'm saying is, if you have an objective function, typically for machine learning, it'll be some least squares, or maybe a log loss and things like that. But let's say that you have an objective function which is a function of the loss function that is being optimized. You can actually use the GAs to optimize on that function. instead. So a correlated, not completely opposing, but a correlated objective function can be optimized using something like a GA. And so for these two reasons, I'm actually going towards the GA in this particular paper. Right? Any questions, please let me know. Yeah?
When we say that S subset has to be the smallest number of features, are we looking at only the number of features or also the variance that is being explained by?
Yeah, so smallest number of features which successfully explains as much variance as possible. So there is a trade-off there, right? Yeah, we decide the trade-off, right? So imagine like using an AIC, for example, right? It's like using an AIC, but then using a GA, right? Yeah. It's non-linear. PCA is only for linear. Yep. And the second thing is, it also tells you, PCA doesn't tell you what the variables are. So basically, if I have an original space, it will give you combinations of variables, which basically give you new variables, right, in the new space. But this actually can give you a original variable space. So essentially, it says that, okay, here is x1, x2, x3, x4, x5, and can use these alone to explain your y. That's great, right? Because I want an answer in the original space. So this is pretty direct in that form. It's not that you can't do it in the PCA space, PCA as well, but it's just that this is more direct. Yep.
Can you give an example of the correlated objective?
Oh yeah. So the correlated objective, let me go back. So the correlated objective function is basically, let me tell you this, right? So I had a objective function which was basically the accuracy of the model, which was from the SVM, plus I actually added a penalty function which actually was the number of support vectors which was being used by the SVM. So in that sense, I was actually penalizing the objective function myself. I was actually adding the regularizing term myself externally. If the SVM wasn't doing itself. So you could do that with the SVM as well, but then we could actually decide on a complex form for that as well. So essentially, if I wanted to be a log form or an exponential form for the number of support vectors, then I could do that. So it's kind of like you can decide it along it. So you can make it a monotonic function of the objective function itself. That would be the best possible case. Otherwise, you go the other direction, then yeah, it's going to get messed up. Yeah. Versus? What is t-SNE? Oh, t-tests. Yeah, of course. So the way we actually do this is that you won't actually be able to get anything out of your t-test when you do this in a single variable way. So what you're suggesting is that can I actually bin the data, for example, or look at the histograms? Okay, that. Yes, you can actually do that. You can do a nearest embedding kind of method, right? But again, when you generate embeddings, you are not operating in the original space. You don't know what kind of functional forms also operate. So for example, the embedding itself has to depend on your topology of the embedding machine, right? So you have an embedding machine, it has several layers. Now it's not a unique way that you can do the embedding. So depending on which, you will get different sets of variables or different set of embedding values for each kind of embedding that you do. Now here is not the case, right? Because I have a deterministic machine that I'm using, which is the SVM. I'm using a deterministic set of variables. I know how exactly they're going to operate. So I have an advantage over that in some sense. So whenever I have to generate embeddings, I'm a little bit jaded about it because of this reason, because it is not unique. Right? If, for example, you generate embeddings and I generate embeddings, totally different stuff, right? And so what I understood what you were saying was that there was another thing which I actually was going to come to, which is that if you actually take a simple chi-square test or a simple linear test, for example, this would kind of shoot that out of the water. I mean, there's no way in hell that can actually even come remotely close to this. But yeah, in terms of embeddings, I don't think that compares really. Yep. And the other thing is that with embeddings, there is a little bit of staticity. So for example, where I've observed this, in this problem, it won't make a difference. But when you're taking financial time series, right, embeddings tend to be a lot more static as compared to something like this. Because the way that I think about SVMs, for example, is that it's like an elastic net with a kernel regression, right? If you take a deep learning machine, it pretty much tends to overfit the machine. It pretty much tends to overfit the data, right? It's not wrong to overfit data as long as the data doesn't change distribution too often, right? It's only wrong when the data keeps changing. And when you take financial time series, all over the place. So that fails again. So for me, this is more ubiquitous as compared to that in subsets. Yep. Yep. Lasso is for linear, yes. It's for linear. If you do the feature engineering, you can use a lasso, yes. It's for nonlinear. Kernels. SVM with kernels is basically so an asymptotic equivalence between elastic nets and SVMs exist in the linear regime. So they're probably equivalent, but then once you get to the kernel, the elastic net it doesn't really do anything. It's kind of a linear, it's only for linear plus L1 and L2, right?
Which features are actually important?
Sure. So decision trees with sufficient number of levels can do it. And again, the problem is that decision trees tend to operate in an ensemble sort of manner. So for me, when I want to do interpolation, right, SVMs are interpolators, very good interpolators. Decision trees are very bad interpolators. If you have if a number of levels which is constantly changing, you want a good interpolation between two levels which is actually sort of nonlinear, SVMs is the way to go. There are disadvantages to using SVMs, which is that it's a complicated algorithm. In terms of computational complexity, it's a lot more. But if I could use it in terms of regression, right, I would not go anywhere else.
Okay.
Yep. All right. So this is these are two cases why I would use genetic algorithms. So the usual GA operates this way, where you initialize a population of genomes. When I say genomes, I basically mean that combination subsets of variables. So if I have x1 to x10, I can have subsets of length 1, 2, 3, 4, 5, or till 10, right? So I can have x1, x3, x5 as one subset, x2, x4, x7 as one subset, and so on, right? So what I would do is basically take all these random combinations in the first generation and generate their fitnesses, right? So I would evaluate the fitnesses of the chromosomes, right? And then I would ask a question, okay, is this converged? And what I mean by converged is it of sufficient closeness to my y variable? Am I regressing it with as much accuracy as possible? And have I hit the number of generations that I want to hit? If the answer is no, what I do is I basically select the top n fittest of that generation or a subset of variables as parents, and I create a new generation from that. I create the new generation, so that process is called a crossover, and then I basically allow for some mutations on that new generation, basically change up one or two variables in each of those chromosomes, and then I again do the same process, right? So let's go over an example, right? This will make it clear. So for example, you have 5 variables. You guys can see the board, can you? Yes? Okay, so basically chromosome 1 consists of variable x1, x3, x5, x7, and x8. Chromosome 2, x2, x3, x7, x9, x10. Chromosome 3, x4, x5, x8, and x10, and so on, right? And these have fitnesses f1, f2, f3, f4, and f5, right? So I have 5 chromosomes. This is a toy example. You have 5 different fitnesses. And what you do is basically the traditional GA takes 2 of these, based on their fitnesses. Let's assume that F1 and F2 are the best of fitnesses. This is ordered in terms of decreasing fitness. And what you do is you choose F1 and F2 as parents, for example, right? So then what happens is that you take 2 parents, you pick a random point where you want to chop them off, and then you join the rest, you create a new chromosome, and voila, you have a new child. So essentially you keep doing this process over and over again, and then you randomly determine that you want to change up. For example, if the child contains X1, you want to randomly change it up to something else. And so again, this is called a mutation. And now the mutated child the fitness of that is evaluated. You go back onto the same cycle again and again. So the thing is that this process, as you can see, is very inefficient because, first of all, you know that you are actually joining 2 parents, right? So imagine the human case. You have 2 humans, right? And you have a fit human, first the top fittest human, the second top fittest human, for example, right? Let's assume this is completely asexual, right? So basically you join 2 fittest humans, you want to take the best features from each human as well. You don't want this to be random, right? And how do I do that? So essentially, if I was going to take it based on, let's call one of them father and one of them mother, just so that, it's kind of obvious, more close to the human example, right? Now what happens is that we want to take the best of both features, but that doesn't happen. And how can we better inform it? So for example, if I have one parent has good pair of eyes, and they're a good pair of ears, right? And the other parent has a good pair of ears and a good nose or something like that. You want to say that the child will have a good pair of eyes, but what makes the child fitter is actually having a good pair of eyes and a good pair of ears, not just a good pair of eyes. So you can see that if I actually take combinations of variables, which is good pair of eyes, good pair of ears, and then good legs, I put this kid out in the jungle, 2 million years ago, he probably would survive. He or she would probably survive, right? As opposed to saying that I just want a kid who has a good pair of eyes or a good pair of ears. So I want this entire system to be automatic. I want the system to actually recognize that combinations of variables are important as single variables themselves. So when I choose the fittest parents, I wanted to actually find out what is the single most important variable. Given the single most important variable, I want to generate the second most important variable, which will enhance that, and the third most important variable, which will enhance that, and so on. So this is the covariance map approach, basically, right? So basically, you're accounting for cross-covariance in multiple dimensions between variables which are already fit. So we'll see how that actually works. So for example, yeah, please.
Can I, instead of creating one child, can I just not add a randomness to the whole process and create multiple child?
You will always create multiple children. I'm sorry. So the way that I explained it was not correct. Typically, what you do is end up creating a generation of children. So let's say that you have 5 parents here, right? What you will do is you will also create 5 children for the next generation. Or you'll create 10 children, for example.
So then in that case, do I need to worry about that, one of the children will not have best eyes and best time?
You don't necessarily need to, but then the amount of time to convergence for the machine to figure that out is going to be a lot. I am basically reducing it quickly. So in fact, if you think about creating that super child, right, in 3,000 generations, I can create it for you in 30. That's the whole idea, right? I wanted to understand that it's not just the features coming from the parents. I also wanted to get a good feature which comes from the uncle or the aunt, right? And that's not possible with the current GA because it basically only takes into account the features which are already present in the parents, right? But the new GA allows for mutations which are beneficial to the kid in such a sense that it is actually beneficial in the rest of the community as well. So that is kind of the second enhancement which I was going to come to, but yes, so essentially this is the idea of creating that super child right now. Now you're kind of in a hurry, think about it that way, yeah? So see, I have always been taught never to put too much text on a slide because it puts everybody to sleep. My way of getting around that was to color it different ways. But this is not the way to do it. I mean, take it from me. I mean, I only did this because if I give this to you tomorrow in a presentation that you can take home, you can actually read it at leisure. But let me actually explain this using an example, right? So let's say that we are going to cross chromosome 4 and chromosome 5. Okay, this is our example, right? Chromosome 4 has X1, X7, and X9, right? Chromosome 5 has X3, X5, X9, and X10, right? So, based on single variable fitness from these two, right, the base fitness of gene X1 is only seen in X4, right? So basically, its fitness is F4, right? Now you come to X3, right? The fitness, it's only seen in chromosome 5, so its fitness is F5, right? So far clear? Now, come to X7. It's actually seen, X7 is seen in both F4 and F5, is that right? Sorry, this is actually wrong. X7 is not seen in both. It's only seen in one. But X9 is seen in both, right? So the thing is that you can say that its fitness is a combination of these two. And let's say that we want to be the simplest possible combination, which is basically an average of those two, right? So it's F4 plus F5 divided by 2, right? And X10 is F5, all right? So this so far is clear. Now, if we start taking combinations, right? X1 and X3. X1, X3 doesn't exist in either of these chromosomes, right? So basically, we give it a nominal value of E, which is actually equivalent to your exploration bonus in your reinforcement learning, if you've actually seen that, right? So E is kind of like, I've never seen it, but I can't assume that its fitness is going to be 0 because I haven't seen it. I'm going to give it a nominal value, right? That's E. But X3, X7 is actually seen, it's actually supposed to be X7 as well, I think here, but it's supposed to be this. So x1, x9 is supposed to be f4, which is basically x1, x9. And here you have x7, x9, which is the important part. I think I skipped having x7 here. That's the only thing. Basically, because it's supposed to be seen here and here, it's supposed to be f4 plus f5 divided by 2. But you get the idea, right? So basically, wherever you see combinations of variables, you're also going to account for their fitnesses. That's the whole idea, right? So the same way, imagine that I can actually create multiple combinations. So this is only 2. I can actually have x1, x3, x7, x1, x7, x9, x1, x3, x5, and so on, right? So the thing is, I create this entire covariance map to dimension about 30. Actually, when I have about 1,000 variables, I have a 2 to the 30 minus 1 size covariance map, which I actually populate. So that's kind of the computational complexity comes from there. So the speed of convergence comes from updating that map, right? But I don't have to, that map updating is actually faster as compared to, I have 5 more minutes, that's what the bell means. But updating that map is a lot faster than actually computing it using an external algorithm. So in that sense, so what I do here is that these 2 chromosomes tell me that this is the fitness, right? But the gene fitness actually comes from all the 5 chromosomes I've seen before, right? So I have seen multiple chromosomes, not just these 2. I've also seen chromosome 1, 2, and 3 before this. So if I were to actually take the fitnesses of something like X3, right? Now, if you go back to the previous slide and look for X3, right? X3 occurs in here, here, and here. So it's actually in 1, 2, and 5. So the fitness of chromosome 3 should be X1. It should be F1 plus F2 plus F5 divided by 3, right? So basically, you have a similar map which you create out of the entire population. So you create a map which is called the base map, and I call this the generational map. And now, for each combination, so for example, if I take X1, X3, I have seen this in the generation with fitness F1, but I have also seen this with a fitness of E in just these 2 chromosomes which are parents. So when I take the overall importance of a combination, it is base importance plus your generational importance, right? So what this means is that I can actually have combinations of variables which are important from the parents. So my parents tell me that my eyes and ears are important, but the generation also tells me that eyes, ears, and not just that, but the nose is also important. So what happens now is I start developing attributes from the entire generation which are also important, not just what my parents give me, right? So this is kind of the covariance map approach which I developed for this particular paper.
Intuitively it makes sense. I'm just trying to understand mathematically how is that happening.
How so? Basically, it's like a Bayesian method, right? So imagine a Bayesian method. So what happens is, step 1 would be choosing the first variable for the child. So first variable for the child would be x1. Let's put it that way, right? Let's say that if you look at all the single variable importances, you do a Roulet selection on this space, right? You have to sum this and this up, right, to get the overall importance. You do a Roulet selection on that space. Let's say you choose x1. Now, given x1 is chosen, what is the next best variable based on the multidimensional covariance map? That is a Bayesian update, right? So it's saying that I've already seen something. Given this, what is the probability of choosing another variable? And given those 2, what is the probability of choosing a third variable? And so on. So it's nothing but it's Bayesian, completely Bayesian. That's about it. So this Bayesian update actually, yes, go ahead.
Are there chances for local optimization if we are choosing one, we are preferring one?
It's possible. It's possible. That's the reason why you actually have to account for a lot of mutation probability as well. So if you account for mutation probability, which takes you outside that local optimization, you're fine. And also when you do GS, you typically what you do is you take a very large first generation. Your first generation should be larger, second smaller, smaller, smaller. If you take a very small first generation, local optimization is unavoidable. Yes. So the amount of exploration is basically because you control the mutation probability. So when you create a new child, let's say the child is x1, x3, x5, the probability is basically of mutating anything is maybe 0.1, like 10%. So you take x1, x3, or x5, the probability of mutating all 3 is 0.1 times 3, right? To the power of 3. Right?
Maybe a very basic question over here. You said that X1 until X10 are the features.
Correct.
And then the F1 to F5 are the fitness.
Correct.
So in a typical case, say for the case which you presented, so how will you calculate the fitness levels?
Using something like an SVM, whatever regressor you want. So imagine that regressor can be your choice. If the regressor becomes my choice, that becomes a genetic program. The regressor is your choice, where you say that, okay, I'm going to use an elastic net, or I'm going to use a logistic regression or an SVM. That's your choice.
So first you calculate the fitness, then
Correct.
you run it through the genetic algorithm.
Yes, exactly. The GA is just used to compute the next best set of parameters given a fitness function on the subset of parameters. Yeah. Okay, so I have 30 seconds, it looks like, so they're shortchanging me on time. Let me go on to the results. So what we did here was on two fronts. We have on astronomy, on machine learning. On astronomy side, we had an unprecedented 99% accuracy on a star galaxy classification and on cosmic distance measurement to quasars. So that's number one. Number two is that we used a very small number of variables in each case, right? I'm going to wait for that bell to go off. Oh, it doesn't go off. Good. So the second thing is that we did cutting edge work in astronomy. So what this does is, instead of using 1,000 telescopes to actually take the data of one particular star or galaxy, we'll just restrict it to like 3 or 4. That way you actually cut down the amount of time which actually goes into this entire endeavor. So you don't waste time. And the second thing is, if you want to measure the distance to a galaxy, which is really, really far away, you don't actually need to do any spectroscopy. Spectroscopy is a very expensive thing in astronomy. So all you need to do is take the measurements in a couple of colors. So astronomical variables are like colors, shapes, and things like that. You just need to take a couple of them and you will have the distance to the galaxy. That is fantastic, right? So in those two senses, the astronomy part of it was great. The second part is the machine learning part, where basically we added feature selections to SVM. So a lot of algorithms don't come with feature selection. So this is one way to actually add the feature selection on top of them. That's one. And the ability to indirectly optimize a correlated fitness function. Again, this is also quite important in a lot of cases. In finance, for me, it's really important because I don't optimize the accuracy in financial algorithms. I optimize something called concordance, which is basically that number of times something goes up or number of times something goes down, and number of times I'm right in predicting that direction, right? So that's the second thing. And then reducing the number of the time or the number of generations to convergence by a factor of 100. So initially I was converging in about 5,000 generations, and now I converge in 100th of that, about 50. So that's kind of, that's the summary of my talk. Thank you.
One more question. So rather than using this as a feature selection method, can I use the same approach to in general estimate the function? So
yeah,
instead
definitely.
of selecting x1, x2, x3 as a combination, if I can randomly select x1 square and into x2 as
okay.
one of the solution,
Yes.
or one of the chromosome,
Yes.
something like that.
This is, what you're describing is called a genetic program. A genetic programming is where you actually choose the functional form which goes on top of the variables also. So for example, I've seen that x1 square works better than x1 to the power of 3, right? So I combine that with x2, I get a better regressor to y, for example, right? Now, when you're actually choosing variables, you're not choosing the functional form, because the functional form is gotten from the machine that you're using, right? The machine, like for example, a logistic regression or a linear regression is a machine, right? All of these are machines. So the thing is that the functional form is decided for you. Given functional form, what is the subset of variables? But if your subset of variables and your functional form are both mutable, that is called a genetic program.
Okay, okay.
Tell us a little bit about the size of the data you were dealing with and
Yeah.
the processing power, convergence time.
Sure. So the number of stars and galaxies, I think I had that on the first slide. I'm sorry about that. Just let me look at that. So I didn't have about 100,000 stars or something like that. And the number of variables was about few thousands, 3,000 variables.
How long does it take to it?
So the thing is that I had parallelized it over about 20 cores. And so it took me about an hour on that. It wasn't slow at all. So the way I did it was that each of the iterations was sitting on one core. And that way, basically, I was crunching to 20 at any given time. 20 cores. 20 threads, actually.